This method can answer key questions about NK cell receptor genetics, such as haplotype diversity and how this relates to disease.
Visual demonstration of this method will help to set up protocol in your laboratory, including the automation process and data analysis.
The main advantages of this technique are that, unlike conventional methods, qKAT, which stands for Quantitative Key Automated Typing, is high throughput and it provides gene copy number.
qKAT can provide insight into disease association. The implications of this technique can be useful for therapy for viral infections like HIV and hepatitis C, in cancer, in stem cell transplantation, and pregnancy disorders. qKAT can also be applied in studying population genetics, as well as exploring expression and functional interactions between KIR and HLA molecules.
qKAT consists of 10 multiplex reactions that specifically target exons of the 2 KIR genes and 1 reference gene. In this protocol, specific primers are designed to detect the alleles of the given gene, and PCR results in short amplicons. After this, dual-labeled specific hydrolysis probes are used to monitor the PCR amplification in real time. Copy number analysis uses relative quantification of the target KIR genes to a reference gene. qKAT is designed and set up to be a high-throughput technique. Hence, each reaction can be performed on just under 1,000 samples. The following steps show the steps involved in the preparation and plating out of each 96-well DNA plate. To begin, after quantifying DNA, dilute the DNA in a 96-well deep well plate.